Hello students, today we'll be talking about translation, a very important process because you have all read about the central dogma concept. The concept says there is a flow of information from DNA to RNA, which is called as transcription, that you have already studied in the last class. And the RNA has to be translated. You have also seen that there is a polymorphism in RNA. That means there are more than one types of RNA. Basically, three types of RNA that you see and come across: ribosomal RNA, transfer RNA, and messenger RNA. All three get together to constitute a machinery for protein translation. So, we'll be talking about translation as such. What are our basic objectives? To define the process of translation, the role of various enzymes, because all biological reactions are enzyme dependent, and also to explain the steps. What are the steps? And normally this is one area from where questions are regularly asked. So you need to have a good amount of clarity. As you can see here out, the whole idea, genetic code, transcription, types of RNA and enzymes that you are already being made aware. You should keep in mind what happens. DNA is transcribed to RNA. In case of eukaryote, it is being processed and then it is being translated. So we'll see every step in details. Let us first of all define polymerization of amino acid to form a polypeptide, in which the amino acid sequence is dictated by information copied in the mRNA. Or in simple words, you can say process of protein synthesis from messenger RNA. As you can see here out, messenger RNA is translated to form polypeptide, which is further converted into protein. So this is the basic idea. There are certain sub-processes that we will get on with. So what are the requirements? To process, we need certain things: messenger RNA, transfer RNA, ribosomal RNA, effectively packed as ribosomes. And you are aware there are two types of ribosomes: 70S in case of prokaryote and 80S in case of eukaryote, where S stands for Swedberg, a pool of amino acid, and an enzyme, amino acyl transfer RNA synthetase. Very, very special enzyme. You need to remember, just like in the previous lectures, you'd have seen Severo-Aqua enzyme, which was instrumental in polymerization of RNA, right, outside the cell. So we have got some factors which we call as initiation factors, elongation factors, and releasing factors, and magnesium ion. You have also seen how this 70S RNA is bifurcated and 80S is bifurcated. There are two subunits, 50S and 30S. And again, 80S has got 60S and 40S. So these are certain things. The RNA polymorphism that I have already talked about. Messenger RNA containing the information from the DNA, which is to be translated. Ribosomal RNA providing a site for attachment of messenger RNA, and also for transfer RNA, whereby we get the amino acid joined in the fashion as coded by the messenger RNA, and transfer RNA bringing amino acid sequentially from the amino acid pool. You find that there are larger subunit and smaller subunit, right? There are three sites, EPA, effectively exit site, peptide site, and amino site. We'll talk about the roles of these sites. So the whole machinery, how does it look like? You see here out. These are the subunits of ribosome, the larger subunit, right? Smaller subunit. So ribosomal RNA and messenger RNA is also attached to the first smaller subunit, and transfer RNA has got also a group here out. So it's an orchestra of all these RNA that enables us to form the polypeptide chain. Transfer RNA, well, you have already seen RNA has got more functions than simply having genetic information. It is catalytic, it is adapter, So transfer RNA molecule have distinctive folded structure, clover leaf structure, as you commonly see it as. So you see this 5 prime, the 3 prime, and so there are certain loops, T loops, right? Anticodon loop, this is a very specific loop because this is the one loop where we have got matching with the code. So it knows exactly where to fit and where to sit. And D loop, each tRNA has got corresponding amino acid attached to the 3' end. This is an important thing that one should remember. 3' end gets that amino acid attached, and there is a process for it. tRNA can read the code on mRNA. How? By interaction of anticodon with a given codon. You see here out, an anticodon loop has got U, C, and A. So it will fit at the place where it gets A, G, and U as per the complementarity. Law of complementarity says U will pair with A, C will pair with G, and A with U again, uracil in case of RNA. And it gets a corresponding amino acid along with it, serine, right? You might find the second one also, AUG, is the anticodon. So what is the codon for it? UAC. UAC. Don't get confused. This is the anticodon. So this will not code for methionine. Rather, it will code for the complementary codon, which is tyrosine, right? Now, what are the basic steps? First, you need to activate the transfer RNA, right? So charging of transfer RNA or amino acylation of transfer RNA. That means amino acid is bind to the transfer RNA. Where does it bind? 3' end. Very categorically, you should remember it's 3' end. It's a 2-step process. The enzyme, as I said, amino acid transfer RNA synthetase, it activates the amino acid. So amino acid binds with ATP to form amino acyl AMP enzyme complex, and pyrophosphate is released. That's the first part. You'll find amino acid is coupled to the transfer RNA and AMP is released. The charged TNM molecule is now ready for use. So how does it happen? The amino acyl AMP enzyme complex, when it comes closer to the transfer RNA, amino acyl transfer RNA is formed, AMP is released. So this is one step. So you can call it activation of tRNA, right? Now you see the ribosome structure. We are considering the eukaryotic structure at this moment, prokaryotic, 50S and 30S. There are 3 points, EPA, exit site, peptide site, and amino site. And how does it grow? Messenger RNA acts as a translation unit. So the RNA that was being formed, it is flanked by start codon AUG and stop codon. Stop codon, as you remember, UAG, UAA, and UGA. Very important, you should remember. I gave a simple code for you to remember: Ullu Aya Gaya, Ullu Aya Aya, or Ullu Gaya Aya. So these are the 3 codons. And AUG, as you find, it's in green. Green says go, red says stop. So green and red, these are the 2 colors that you can keep in mind. AUG has got a dual role. Already discussed. What is this dual role? It codes for methionine as well as it starts the process of translation. So it is the initiator codon. There are some UTRs found on either seed. That is for effective translation, untranslated region. So steps of translation, as you can find in every race, there are 3 steps. Initiation, how you take off; elongation, how you move in a relay race and give the baton to the next participant; and termination, the finishing line. 3 stages. Initiation, what happens? In crisply words, you can say ribosome binds mRNA at start codon. Elongation, the ribosome moves 1 codon unit every time and brings in a new amino acid and joins to the existing one, thereby making a polypeptide chain. And termination, where stop codon is reached. So now the ribosomes separate and the process of translation stops. Before we go with initiation, let us talk about initiation factor. So first of all, initiation factors in prokaryotes, there are IF1, IF2, IF3, and in eukaryote there are more than 3. There are around 9 factors: EIF1, EIF2, EIF3, EIF4A, 4B, 4C, and 4D, EIF5, and EIF6 in eukaryotes. Initiation factor binds to the smaller subunit, right? You see here out, initiation factor binds to a smaller subunit of ribosome, mRNA binds to a 30S ribosomal subunit, and AUG codon lies on the P site, and the second codon lies on A site. You can find here out P site and A site, right? So methionine carrying transfer RNA is placed at P site, and 50S subunit binds with 30S, forming the complete unit. So translation initiation complex, this is called as translation initiation complex. It is ready to now invite other tRNA to bring more number of amino acid. But there is a question being asked normally: how does it recognize where to bind, right? So in mRNA, you have already read about UTRs, untranslated region. Why it is UTR? It does not take part in translation, but it has got some sequence that allows in recognizing where these mRNA will bind with the rRNA. And there are some sequence in mRNA which is known as Sine-Dalgarno sequence. It binds with rRNA, ribosomal RNA, and hence it is about to start the process of translation. Hence in your book it is given, for efficient translation process, UTRs are needed. Elongation, second stage. So tRNA carrying next amino acid comes into A site, right? Now amino site, there is a peptide bond formation as because this tRNA are charged, right? So when they leave at E site, there is a bond formation between the two amino acids. Ribosome moves one codon ahead. There is an enzyme translocase that pulls it. One codon unit means 3 bases, right? And the direction remains 5' to 3'. This is the direction of translation. Now, first tRNA exits from E site, right? The amino site RNA comes to P site, and amino site gets free. Thereby, next amino acid is invited, right? So you find here out, you see this, there is a movement. This is A site. At peptidyl site, new amino acid coming, it is moving one step forward, and thereby another vacancy is created, just like a musical chair game. The amino acid in the P site breaks the bond with its transfer RNA, forms a peptide bond with the amino acid in the A site. You'll find the process of elongation, right? And there is a direction of translation. You find the ribosome attached to the mRNA and whole number. So there is a polyribosomes being cited. Many copies are to be formed. Now the termination. tRNA do not have anticodon for stop codons, right? We know that it's anticodons that has got matching with the codon. So tRNA do not have anticodon for stop codons. If stop codon appears on the A side, the tRNA fails to bind, right? It fails to bind because there is no anticodon. Now a protein called as releasing factor, RF, binds to ribosome and causes dissociation of the two components, 30S and 50S in case of prokaryote and 60S and 40S in case of eukaryote. And you find the termination factors in case of prokaryotes seems to be RF1, RF2, and RF3. And in case of eukaryote, it is ERF1, right? Now, termination factors, what happens here out? See, as soon as the release factors are attached, there's a dissociation of two subunits, and the process of translation stops. This is the whole cycle that you can see. Start codon reaches initiator site at the A site, or I'll say comes to the P site, then joining of larger and smaller subunit. Another transfer RNA comes, pairs with the next codon beside the initiator RNA, and then amino acid on initiator transfer forms a peptide bond with the amino acid. There is a movement of this whole transcription unit by one place so that there is a shift of place, and one on P site moves to A site, E site, and A site moves to P site, and A site becomes free until next transfer RNA brings another amino acid. So this is what we have read, the process of translation, process of polymerization of amino acids, but it's a heteropolymer. It requires 3 types of RNA, mRNA, tRNA, RNA, amino acid enzymes, and several protein factors. mRNA carries information from DNA, which is to be translated into protein. Ribosomes acts as a cellular factory for translation as well as catalyst in case of prokaryote. And you know, the whole process can be summarized as 4 important steps: activation, initiation, elongation, and termination. Activation, charging of transfer RNA, initiation of protein synthesis, during which both subunits are joined together and starts acting like a factory, and initiator codon being AUG, elongation of polypeptide chain, whereas more amino acids are brought in and joined, and termination when the whole system reaches the stop codon. There is no anticodon in transfer RNA corresponding to nonsense codon, hence the process stops. So this is what you need to understand and remember, and then you can refer to the NCERT, read it, and you can write the concept all again. Thank you. See you in the next video with more concept, regulation, gene regulation.